Okay, so let's start again. We discussed about respiration, then we discussed about fermentation, which is an anaerobic part of respiration. And then we discussed that in industrial biotechnology, every product, the process through which we produce this product industrially, generally we call it fermentation, even if it involves oxygen, even if it involves aerobic respiration. And like we do experimentations, culture, and grow these cells in labs, we use flasks. Let's talk about liquid cultures at the moment, not about the agar plates or the semi-solid or just solid culture. But when we do this on a commercial scale, because we are producing for thousands of peoples, if not millions, these flasks are not good enough. We need to go like some kind of a flask that could have 50,000 flasks volume in that one big flask. And that one big flask, we call it a fermenter or a bioreactor. This is a general structure of a fermenter or a bioreactor, you could see. And it is the heart of fermentation or the bioprocess technology. So you know now fermentation technology, bioprocess technology, or industrial biotechnology. These are, or even in some cases, you would see chemical engineering. These are all interchangeably used, as well as the name of fermenter or bioreactor is actually used interchangeably. So a bioreactor is a fermenter is a bioreactor. Generally, it is used like that. It's an enclosed vessel. It's basically a device in which we cultivate our organisms, our cells, if not organisms, in case of using mammalian cells for some desired products. We would call them cells. If we are using some bacterial cells, then we could call them microorganisms. Even if we're using fungal cells, we would call them microorganisms. So it's an enclosed chamber, and it is actually confined chamber as well. So a confinement system for the cultivation of cells, where we provide, it is designed in such a way that it provides the right environment for the optimal growth and metabolic activity of the organism, because we want the organisms to be happy. If they are not happy, they will not divide rapidly, and if they do not divide, their cell expression, gene expression would be slow, and we do not want that. We want them to be metabolically active, having plenty of nutrients, so that they divide and they produce our desired product, either through the expression of a gene of interest or just normal, their primary or secondary metabolites. Technically, there is a difference between fermenter and a bioreactor. When we use prokaryotic cells like fungal cells, not all fungal cells, but if you're working with yeast cells, Saccharomyces cerevisiae, Pichia pastoris, Schizosaccharomyces, Aspergillus niger, these eukaryotic cells, microorganisms, when we are culturing them, we call the confinement system a fermenter. But when we are using eukaryotic cell, like insect cells, like mammalian cells, then we call it a bioreactor. As I told you, these names are interchangeably used, but technically there is a difference. Different types of bioreactors: continuous stirred, airlift, fluidized bed, packed bed for a bioreactor, membrane bioreactor. We do not need to go into the details of these bioreactors, but just a small idea about how bioreactor is enough. Typically, a bioreactor, this is the typical structure of a bioreactor or of a fermenter. So there's always an agitation system so that all the things are getting mixed. And this is very important because for aeration to this inlet, we will have we will be introducing oxygen. As I told you, this we will call this fermentation, but there will still be in some cases oxygen involvement. And then the medium, sometimes it's a continuous batch chemostat in which we will be continuously providing medium, at the same time continuously removing medium. So these inlet and outlet, the mixing through these rotors, agitation system. Then there are a couple of monitoring systems, because we want to have an eye on the acidity of the medium. We want to have an eye on how much carbon dioxide is released. We want to have an eye on the temperature. All these things so that we can carefully regulate all these processes and provide the best possible conditions for the growth and the division of these cells. These are very important, some very important parts of the typical fermenter or typical bioreactor. And that the sterilization and maintenance of sterility is very important, because if we design a medium for product X through Aspergillus niger, and in the medium there is a contamination of, let's suppose, Klebsiella or some other bacterium or some other fungal cells, it's not good enough. It's not good. It's not good news. So we have to make sure that we are working in sterile conditions, and we are working only with the only cell that we need it to be there. Industrial fermentation, also called as wide biotechnology, in which living organisms or their microorganisms or their products on a large scale are used, are exploited for the synthesis of commercial products. Commercial product could be therapeutic or non-therapeutic, could be food products, could be additives, could be a number of other things. For example, beverages, additives, supplements, healthcare products, antibiotics, enzymes, biomass production, biofuels, all these are involved in, are included in fermentation technology, industrial fermentation. One common example is ethanol. So ethanol, it's a very classical example for fermentation biotechnology. Since we are living in a Muslim country, we do not consume ethanol or alcohol, as it is banned by our religion. But ethanol is used in a number of processes. For example, look at the current scenario of Pakistan. It is because of ethanol that within, I would say, not more than 2 weeks of the pandemic of coronavirus, SARS coronavirus 2, that Pakistani industries started producing hand sanitizers, and there's plenty of them around now. In the first 2 weeks, there was a lot of problem. One of the major ingredients of these hand sanitizers are alcohols, ethanol or methanol. But methanol is not that much used because it's not as effective as ethanol. Ethanol everywhere. Now these days, ethanol is everywhere. It's like gold these days for all over the world, because it's because of its use in sanitizing. The other biggest use, it's a solvent for a number of chemical reactions, and you can also use it as a fuel. Well, that's a very sophisticated technology. You have to change the engines of your cars, but that is a viable solution, one of the viable solutions to sustain, to have the sustainable development, sustainable goals. You can use the corn stores, or you can use the corn, and then once you brought them to the industry, you have to mill them, and you have to cook them so that all the sugar in the ground material is dissolved, or the starch or the cellulose. It is converted into sugar. Why? Because these yeast cells, they would thrive on these sugar molecules, and eventually after saccharification, which is the conversion of all these organic molecules to sugar through cooking, would be introduced into a fermenter. And after the fermenter, we will culture them, we will provide them all the important nutrients, maintain the pH, and in this case, we would have no oxygen involvement. So we will be pumping through all the time nitrogen inside, so that we ensure there is no oxygen. And eventually, when the process is completed, we will distillate it. And after distillation, we will have the ethanol, and then ethanol can be transported for any purpose. You can use it for any purpose. The next topic of industrial biotechnology is antibiotics production. How do we produce antibiotic? It's similar to fermentation process that we discussed for ethanol, yet a little different. Here, it depends on the microorganism, but mostly it will involve oxygen, because these actinomycetes, which are a class of bacteria very famous for biosynthesis of antibiotics, or the original Penicillium notatum, or the better strain Penicillium chrysogenum. They are all aerobic microorganisms, so they will need oxygen for their survival. So we will have a medium product preparation. Then we will sterilize the medium through heat. Fermentation will be done in a fed batch mode. A fed batch mode, it means you have all the nutrients, either one nutrient or more than one nutrient, and you introduce into the fermenter, and everything will be there until the end. There will be no removal of a medium, and there will be no addition of new medium, because this is the best conditions where the antibiotics are produced. As antibiotic is a secondary metabolite, it will be released into the medium. The seed culture, once you have the fermentation, you have the medium, you need a seed. The seed will be a few hundred or a few thousands of cells of bacteria or fungi, depending on which one you are using. In this case, penicillin, so it will be penicillium spores. And then after introduction of them into the fermenter, they will start dividing and producing their primary and later on secondary metabolites. One of the secondary metabolites is our candidate of interest, that is the antibiotic penicillin. We will have to remove the biomass. There are many methods for the removal of biomass. Rotary vacuum filter is one of them, which is commonly used, and the pH has to be maintained close to neutral, because this is the optimum pH where these bacteria, these fungal cells love to grow. In case of adjustment, we can use phosphoric acid for the pH regulation. Similarly, when the biomass is removed, we want the penicillin to be dissolved in the filtrate, and for this purpose, a number of organic solvents could be used, for example, butyl acetate. The next step is the centrifugation extraction, because we want the solid and the liquid to be separated. So density gradient extraction is used, and the penicillin will be dissolved. Because of the addition of butyl acetate, all the penicillin antibiotic would be actually dissolved in the liquid, so supernatant. And then after this, we need to dry, because most of us know that antibiotics comes in coated tablets. It's very important to remove the moisture because when it's dry, nothing can grow in it, and it has a longer shelf life as well, and it's easy to package into so that it could be sent to the consumer market.